Module 2 is all about the cell. We'll start a little bit on the history of cell theory, this whole idea about the fact that all living things are made up of cells. But we'll spend the bulk of this module talking about the cell itself, right? And it's two major cell types, the prokaryotic cells, which is going to be the major focus of our semester, and then the eukaryotic cells as well, right? So fun stuff, I promise you. Let's do this. So, modern cell theory states that all cells come from other cells. It's this whole idea of biogenesis, right? So, a long time ago, there used to be a prevailing theory of this thing called spontaneous generation, where cells just sort of came up, right? And based on the observations, based on the knowledge of people at the time, that was kind of like the best explanation that they had, right? So your book goes all into that and all this whole proving and then disproving of this idea of spontaneous generation, right? But now we know that cells don't just come from nothing. They come from other cells. And then it's also very well established that cells are the fundamental units of living organisms, right? So all organisms are made up of cells, and cells are our smallest unit. Right? And so we are a collection of billions of cells put together. And of course, we know and we will learn that there's much life that can be sustained at the single cell level. The term cell itself was coined by Robert Hooke, who did some microscopic observations of cork, not even cells, like not even living things. It was cork. And then he noticed that there's these little squares, right? They kind of reminded him of monk cells, like monks live in cells, I guess, not jail cells, but that's what they call them in monasteries. And he says, oh, look at this. This is made up of cells, right? Wasn't even really referring to the fact that living things were made up of cells. It was only a little bit later when Matthias Schleiden and Theodor Schwann were looking at plant and animal cells. And then they realized that all these tissues were made up of things that looked very similar to what Robert Hooke was looking at. And then so then they adopted the term that he used, cells, right? And then so that's where the term cells started to refer to the fundamental units of living organisms. All right, so it was kind of established around this time then that living things were made up of cells, right? But this whole idea of where they came from wasn't quite understood, right? In fact, again, many people thought that they just sort of came up, right? And it wasn't until Rudolf Virchow, he wrote in his seminal work, Omnis Cellula a Cellula, which means all cells arise from cells, right? And basically, every single cell, the origin of cells was the division of pre-existing cells. Interesting story with this guy. Him and this guy, Robert Remak, were kind of talking about stuff, and Robert Remak actually wrote that the origin of cells was the division of pre-existing cells, or at least he thought that's what it was, right? But he didn't have any hard evidence. He really didn't have a strong conviction other than just that's kind of what he thought, right? And Rudolf Virchow initially said, you know what, I don't think, he said some cells do that, but I don't know if all cells do that, right? But then upon more work and more research, he realized, you know what, this guy is right. So then he ended up writing this thing and took the idea for his own, right? So oftentimes it's said that it's borrowed the ideas from Remak, but in many ways he took Remak's work, he built on that, and then he did not credit Remak, who was kind of pissed off about that, of course. The other interesting thing about the story is this term omnis cellula a cellula. It's not even a phrase that he coined, right? It's this French physician, Francois Vincent Raspail, or something like that. He wrote Omnis Cellula a Cellula way back in the day, not that long ago, not long before this, right? Just kind of as this interesting phrase, right? And Rudolf Virchow, he kind of liked that phrasing, right? So he coined that, or he used that phrase, and then now often this phrase is attributed to Virchow when it wasn't him that actually wrote it. So around this time then says, okay, cells come from cells, right? Which then, of course, means that we knew of the existence of these living cells, right? We knew of all these kinds of things. And then so around this time, cells started to be attributed to as well as disease, right? This whole idea of the germ theory. And remember, before this, there was this whole miasma theory, right? This theory that disease comes from quote unquote bad air. If you're in the same room as someone who is sick, they're breathing or exhaling this bad air, and then you breathe that in, and then you yourself get sick, right? I mean, it's to some extent you can see where some of this theory has come from, right? But the reality is it's not quite that simple, right? And so The germ theory of disease was proposed, around 1500s, right? But it wasn't really widely accepted, right? This whole idea that it's these little microscopic things that we cannot see that causes disease, right? But then there's this physician, Ignaz Semmelweis, and this is in the mid 1800s, right? He was a physician, and then he noticed that physicians were often the cause of infections of patients. And actually, back in the day, hospitals a terrible place to be in. Many people died in the hospitals. Many people got sick because of the hospital. And there's stories of people aiding in the birth of a child, and then they didn't know what to do with the afterbirth, so they just kind of stuck it in their pocket and they just walked around with that, right? Or they'll have pens or pencils. I'm not sure. Did pens exist back then? I'm not sure. But they'd have writing utensils, and then they would poke people with them, and they say, hey, check out this thing and check this thing out, right? And they themselves did not realize that they were causing much of the spread of disease, right? So Semmelweis, he had this ingenious idea. Hey, you know what? Let's wash our hands before we touch patients, right? It seems so simple. Yet, believe it or not, his work was not well accepted. And there were many people who said that, you're crazy, man. We're doctors. We don't cause disease. It's other things that cause disease, right? So it actually took a little while for this to catch on, believe it or not. Sometime around this time as well, Jon Snow, not the famous person hanging out at the wall, protecting us from the White Walkers, right? This is a different Jon Snow. He is considered the father of epidemiology. There's this major cholera outbreak in London. And then he, through a series of investigations, tracked the outbreak of cholera to a well. And once again, it wasn't this whole idea of, well, we're just breathing this bad air. He found the root cause for where this came from, right? And we'll talk a little bit more about Jon Snow's epidemiological study later this semester, right? But these things were now starting to let people understand that, it's not just bad air, not some sort of vague reference to things. There are actual causes to disease. And by closing off the well, by washing your hands, we can actually stop the transmission of disease. So then building on these things, Louis Pasteur, one of the founding fathers, right? One of the most central figures of microbiology, who really has his hand in so many things, right? He hypothesized, or I guess he made some observations that microbes spoil food. And there's a real cool story about this, and maybe one day if you're in my lab class, I'll tell you all about it, right? But he learned that microbes can spoil food. So then he thought, hey, you know what? If they can spoil food, that means they can also cause disease, right? So he kind of made that connection. So then there's this physician, Joseph Lister. He said, hey, you know what? If that is true, if microbes can spoil food, and we know how the microbes get in the food, right? It's through our tools that we use. It's through the containers that we use. And then if microbes can cause disease, what if whenever I'm cutting open somebody, I can introduce germs or bacteria or these invisible things, microscopic things to the patient and making them sick? So he developed procedures for sanitizing everything using carbolic acid or phenol to sanitize the hands, the tools used, and then once you're finished, to clean those things off. And then again, his work has really dramatically reduced the incidence of people getting post-surgical infections in the hospital. But once again, his work wasn't immediately caught on. It took a little while, right? And there were these people that they called Listerians, the weirdos who sanitize everything before surgery, and then the ones who did it the normal way, right? Interesting fun fact, right? So Lister attributes his name to, Listerine, attributes his name to Joseph Lister, right? Because of the fact that early formulations used carbolic acid or phenol in Listerine, right? So whenever you see Listerine, you think of, the person that decided we need to sanitize everything before we cut someone open with it. And then finally, this is probably towards the late 1800s, Robert Koch established this protocol or postulates to determine not just that microbes do cause disease, but which microbes cause disease, right? Because again, around this time, it was kind of clear that microbes do cause disease, right? But how does one say, oh, well, it's this one that's causing this disease, and it's that one that's causing that disease, right? So he has these things that we call Koch's postulates, right? That determines how a microbe causes which disease, right? This whole theory of one microbe, one disease. All right, and then with respect to cell theory, one of the most recent developments was this whole idea of this endosymbiotic theory. Right, this idea that, and we'll talk about what they are, the mitochondria and the chloroplasts, they arose from this symbiotic relationship between prokaryotic and eukaryotic cells, right? So, this idea that long time ago we have our early eukaryotic cell, and along the way it decided that it's going to eat this bacterium, which is of course what eukaryotic cells would have done. They would have eaten this early ancestral prokaryotic cell, right? But along the way, this bacterium was able to survive, right? And then so because it liked the protection of being in this eukaryotic cell, it's much warmer and it's not so scary than it is out in the outside world, right? And of course, then it's doing its whole metabolism thing. And this eukaryotic cell said, hey, you know what? This little thing inside me is making so much energy that it doesn't even need it all for itself. So then I don't have to work so hard to get energy. All I need to do is a little bit to survive by myself. And then over time, these things kind of became not even symbiotic anymore. It just sort of became a part of that cell, right? And similar goes for chloroplasts. And, much of the evidence for this is the fact that both mitochondria and chloroplasts, they replicate independently. They don't wait for the cell to replicate. They just kind of copy when they need to be copied, when they feel like copying themselves. They have their own DNA, right? Why would they need their own DNA? But they do. And then they have their own ribosomes. And we'll talk a little bit later about what ribosomes are, right? But they're able to do these things, almost live separately, but yet together, right? So that's this whole idea of endosymbiotic theory. All right. So that's a little bit about kind of the history of cell in a very short time. Probably a little bit longer than it should have been, but a little bit of history of cell theory. So let's just jump into what cells are, right? So, and this is going to be a theme for just about everything in biology. Cells, we always say this, are incredibly diverse, right? So many different sizes, shapes, structures, functions, right? And then we often look at like our typical circular-ish cell, right? But the truth of the matter is cells are just incredible shapes and just all these different types of things, right? And then overall, though, we can say that with relative confidence, right, that all cells have a plasma membrane, sometimes called a cell membrane, a cytoplasm, which is basically just the liquid inside, right? You can almost think of it as like the blood of the cell, right? Sure, why not? Let's just call it the blood of the cell, right? And then chromosomes, which is basically the genetic blueprint, i.e. DNA, and then ribosomes. And ribosomes are used to make proteins. And we'll actually talk a little bit more about each of these as we work our way through these through this chapter, right? Or this module, right? But it's these four things generally that are required for one to be considered a cell, right? Two major cell types, prokaryotic cells, eukaryotic cells. We kind of talked about these already, with our major distinction being that prokaryotic cells have no nucleus, eukaryotic cells have a nucleus, right? Again, a lot more differences in that. And I did promise at some point we will explore the differences, right? So guess what we're going to do? We're going to start exploring those differences, right? So if we were to have a checklist, right? And again, we're going to go these general broad strokes, right? All prokaryotic cells have one circular chromosome that is not in a nuclear membrane, right? And we're going to define some of these things as we go, right? But basically, one chromosome that's not enclosed in a membrane, right? Whereas eukaryotic cells have a linear chromosome, right, in a nuclear membrane. And this is what we call, did that not work? There we go. Oh, this is going to be fun, right? This is what we call the nucleus, right? So the nucleus is this compartment in which that's wrapped, this membrane-bound compartment which encloses the linear chromosomes, and usually multiple chromosomes. Prokaryotic cells don't have histones with their DNA. I don't know if we're ever going to talk about histones in this class. Oh, we might. Eukaryotic cells do have histones, right? And it's kind of an important organization, and that's why the eukaryotic chromosome, which is so large, can seem incredibly small. Prokaryotic cells don't have organelles. Eukaryotic cells do have organelles. And prokaryotic cell division is very simple. It's this thing called binary fission, whereas eukaryotic cells have this process called mitosis. And in fact, there's another one called meiosis, which is a specialized form of cell division, right? So we're going to start very basic, just broad, one circular chromosome, many linear chromosomes. No histones, have histones. No organelles, have organelles. Binary fission, mitosis, right? So we'll just keep it like that. And of course, as we start to really unwrap the layers, we're going to start to see some more significant differences, okay? A little bit more about prokaryotic cells. We talked about some of these already. Again, many shapes, many sizes, right? They are relatively small, right? Much smaller than eukaryotic cells, right? The average size is 0.2 to 2 micrometers in diameter, 2 to 8 micrometers long, really depending on the cell type, right? What does that mean exactly? They're small. Okay, that's really what it is. They're small. Not as small as viruses, but definitely smaller than eukaryotic cells, right? And so that's one of the defining features. If you're wondering, hey, is this cell that I'm looking at prokaryotic or eukaryotic? Stick it under the microscope, right? And if it seems small, it's prokaryotic. If it's larger, it's eukaryotic, all right? Of course, if you don't have a comparison point, how do you know it's small? But I digress. We talked about the 3 basic shapes already, right? Coccus for round cells, bacillus for rod-shaped, curved, that has some sort of spiral or twist, right? Sometimes we don't really know how to memorize these things, right? So this is what I often use. Coccus cells, notice it's an O there, right? So round cell for coccus. Bacillus, right? The L's are kind of dominant in bacillus, right? Those are rod-shaped, and they kind of look like L's, right? Or rods. And then curved, of course, just has some sort of curve. So you can have a very simple vibrio, which is like a boomerang or one little curve here. We can have spirillum or spirochetes that have like these many twists and shapes and that kind of stuff. And there are many other shapes. There are square bacteria. There are pyramidal, right? A lot of interesting shapes for microbes. Many of the weird shaped ones tend to be archaea, right? But for the most part, the kind of stuff that we're going to see are always going to fall into these, and then sometimes are curved, right? So it's the coccus and bacillus that are probably the most common. Okay, so if you feel like it, now will be a good time to take a break, right? Kind of gather your thoughts about what you just learned, right? Because we are now going to jump into the prokaryotic cell, right? So we're going to focus on the prokaryotic cell. Along the way, we're going to learn a lot about these structures, right? And so here we have, and the books always show this, right? They show here's a prokaryotic cell, right? So what I've done here is, actually I call this our typical prokaryotic cell, right? And we're doing this in parentheses because this is not a typical prokaryotic cell. This is some uber super duper prokaryotic cell that has incredible numbers of structures that cannot be found in every single prokaryotic cell, right? So this is like our enhanced super duper prokaryotic cell, right? But it is a nice picture that shows many of the various structures that you would find. We're going to take this path going from the outside of the cell to the inside of the cell. Okay, so with that then, let's begin, right? As we start to peel through the layers, we will start with our outermost layer, the capsule. And this is a structure that is not found in all cells, okay? So how do I want to indicate this, right? Let's just call it not. And when I write that, that means not in all cells, because I don't want to write it all out. All right, so we're good with that? Thank you. So the capsule, aka a glycocalyx. So the glycocalyx is the broader term, right? Glycocalyx singular, glycocalyces plural. The term glycocalyx refers to a sugar coat, right? It's on the surface of bacteria. Sugar coats are made of, guess what? Sugars, right? And what type of molecule are sugars? Thank you, carbohydrates, right? So they're made up of these carbohydrates. And, carbohydrates tend to be kind of goopy, right? Kind of like sticky and slimy. And sometimes they're called S layers because of their resemblance to slime layers. Interesting enough, when we stain bacteria that have these glycocalyces or S layers, we actually can't color the S layer because of the fact that there's really nothing for colors to stick to. So we often do what's called a negative stain, right? We can stain the background, we can stain the cell, and then it's this clear white outside, this border, that is the capsule or the S layer or the glycocalyx, right? This we see it by not seeing it. Because of its stickiness, right? It's used to do a number of things. We can use it to adhere, which means to stick, right? Maybe I want to write that in there. Do I want to write that in there? Let's do that. All right, so we can use that to stick to surfaces. We'll learn later that it can be used to form this thing called biofilms. It helps prevent microbes from drying out, protects it from ingestion, right? Let's maybe define that as well, right? Which means to be eaten, right? So it prevents our immune system cells from eating them. It can inhibit antibiotics because the antibiotics won't be able to access the cell, right? And disinfectants as well, right? So it provides this overall thing. Again, each specific glycocalyx is going to have a specific function, right? Or a feature. So they're not all necessarily characteristics of every single glycocalyx, right? But in general, these are the types of things that a glycocalyx can offer a cell. Two general types: the capsule, which is a bit more organized, very firmly attached to the cell, versus a slime layer, which is a little bit more unorganized, loosely attached, right? Still made of carbohydrates. They're still both sugar coats, right? It's just the organization is slightly different. Capsules can be important in virulence, which is this ability to cause disease, right? So in microbes that can cause disease, right? So for example, this one here, Streptococcus pneumoniae. Streptococcus pneumoniae only causes disease when it's able to make a capsule. And there are strains of Streptococcus pneumoniae that don't make a capsule. And they, in turn, are not virulent or not pathogenic, right? So, and again, this in case would then be because the capsule protects it from our immune system, protects it from being eliminated by the immune system. S layers, on the other hand, again, a bit more unorganized, more loosely attached, right? It's just this kind of this sugar coat that's on the outside. It's usually on the outside of the cell, but some archaea can actually use it as a cell wall. So that for them, that's kind of like their cell wall, as opposed to bacteria, which we will learn have a different type of cell wall. Generally, they're not well studied, right? They're often in the wild. Oftentimes they probably are involved in things like biofilm formation, but definitely / probably help with protecting the cell, help with adherence and things like that, just based on the general nature or characteristic of the slime layer itself. Okay, so moving on. Let's talk about these various what we call filamentous appendages, right? And again, these are not common to all bacteria. So only some prokaryotic cells have these various appendages. And it's not like if you have one, you have them all. Some will have only FIMBRIAE, some will only have a pilus, some will only have a flagellum, right? But there are various protein appendages stuck to the outside, and each of these are actually very different in function. So even though they look similar, they are these long or short hairs that are sticking to the cells. They all have specific function. Okay, so let's move in and talk about FIMBRIAE and PILI. And even those that look so similar, they actually have slightly different functions, right? So FIMBRIAE are used to stick to things, right? So they are these, let's see if I can get this, these short little hairs that are sticking out here. These guys are FIMBRIAE, right? And so they're used to stick to things. So for example, Neisseria gonorrhea, guess what disease it causes? That's right. Sticks via these FIMBRIAE, right? So they're able to stick to things, right? E. coli, pathogenic E. coli has FIMBRIAE that's able to stick to our intestinal linings, right? So many of these microbes, these pathogens that are able to cause disease, use FIMBRIAE to stick around. And even non-pathogenic microbes, they will also have FIMBRIAE that allows them to stick to cells, right? Adhere to various surfaces. PILI, on the other hand, are used to transfer DNA, right, via this process called conjugation. And we'll learn about conjugation a little bit later in Module 7, I believe it is. I don't have my chart with me, but I believe it's Module 7, right? So we're going to learn about conjugation and how the pilus, singular, right, can be used to transfer DNA. And then some PILI can also have some sort of limited motility. Motility means the ability to move yourself, right? It's not amazing motility, but it's some sort of motility. But for the most part, if we're going to just dumb it down, right, we're going to say FIMBRIAE are used to stick. The pilus is used to transfer DNA. And then that would be sufficient for us with respect to these structures, right? Now, when we're talking true motility, we're talking about this thing called a flagella. These things called flagella, which is plural, or flagellum, singular, right? So we have a flagellum is able to give bacteria this thing called motility, right? This ability to move. right? Made up of three basic parts. We have the filament, and that's the part that's on the outside of the cell, right? This long filament. The hook, which is kind of like the part that attaches to the basal body, which is the motor, right? To be honest, that's about as much as I want you to know. Of course, there are many, many parts to this thing, many types of proteins involved, right? But, if we just get the three basic parts of a flagellum, that will be sufficient. More I want us to understand is how a flagellum works and its significance to the cell. Now, bacteria can arrange these things in many different ways, right? And so we refer to bacteria that have flagella via the way that the flagella are arranged, right? So you can have a single flagellum, and in this case, you will be monotrichous, right? So we say, oh, this is a monotrichous bacterium, right? It's because it has one single flagellum, as opposed to an amphitrichous, right? Remember the word amphipathic, both hydrophilic and hydrophobic, right? An amphitrichous bacterium has one single flagellum on each end, okay? Notice the word trichous, that pretty much refers to a flagellum, right? So monotrichous, one singular one, amphitrichous, one on each side. Lophotrichous means multiple flagella on one end, okay? Sometimes actually lophotrichous can also refer to cells that have multiple flagella on both ends, okay? So sometimes you'll see that as well. But basically it's a bundle of flagella on an end of the cell. And then peritrichous cells are cells that have flagella all over the cell, right? So here they have, they're completely covered in flagella, but when they swim, they're going to go and move all the flagella in the same direction. They're all going to work together to get this thing to go and swim that way. And I guess there's a fifth characterization, right? Cells that don't have flagella, right? And we would call these things atrichous. Right, the prefix A referring to without, right? So atrichous bacteria are cells that don't have flagella, and most cells are atrichous, okay? So how do they work, right? So flagella are relatively simple but ingenious, right? So they spin the hook in a certain direction, right? Which then allows the flagella then to go and rotate a certain way. And then, so when we rotate them in a counterclockwise direction, all the flagella start to stream behind the bacterium, right? Causing the bacterium to move forward. And we call that a run or a swim. It's a continuous movement in one direction, right? And it just goes straight. It doesn't turn, right? So bacteria, they don't turn, right? They just go in one direction. When we spin these things in a clockwise direction, right, the flagella then kind of flail out to the side like we see over here. Right, it's all like, woo, let's just spin everywhere, right? And then what ends up happening is because the flagella are everywhere, the bacterium just tries to go everywhere in one direction. It causes this thing called a tumble. So this thing will actually go blah, 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 like that, right? And it's just a basic random change in direction, right? So when we put this thing together, this is how a bacterium would swim. Here's a counterclockwise rotation, it's a run, la 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 la, and then just at some point it'll flip and say, whoa, let's spin around, right? And then it's like spin the bottle, we're gonna spin, and then now it's pointing in this direction. Now it's gonna run, and then we're gonna spin, and then we're gonna run, and then we're gonna spin, right? And just these things happen at sort of random intervals, right? It actually cannot control how it cannot control the tumble, the direction that it wants to go to, right? But what bacteria can do is they can try to inhibit the tumble from coming for as long as possible, or they can get it to tumble sooner than it wants to. So it has this internal clock. At some point, it has to tumble no matter what, right? But it can try to hold off as long as possible, right? Or if it doesn't like where it's going, it's like, oh, I don't like where I'm going. I'm just going to tumble right away, right? So it can kind of do that as well, right? And so this ability is called taxis, this movement towards or away from an environment, right? So let's say there's an attractant right here, right? An attractant is something that the bacterium wants to go to, right? We can do something called positive taxis, right? So in this case, here's our run, and look at this bacterium is swimming away from the attractant. It's like, oh, I don't like this, right? So I'm going to induce a tumble. Bum, 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 bum. It tumbles, right? And starts swimming this way. It's like, okay, this is not bad, right? Not quite where I want to go to, so let's spin again. Bang, 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 right? Now it's like, oh, randomly, hey, this is the direction that we want to go to. We're going to have a long run. At some point here, it's going to once again tumble and then start swimming in a different direction, right? But over time, through these random changes, it'll start to move and kind of hang out in the area that it wants to, right? Based on this ability to control the length of when the tumble is induced, right? Similarly, we can have something called negative taxis, movement away from an environment, or repellent. Sometimes there's a toxin, or there's things it doesn't want to be around. It's like, you know what, we're going to swim away. And then now the opposite would be the case. When it's swimming in a direction away from the repellent, it'll try to make us run as long as possible. When it's swimming towards a repellent, it's going to try to tumble as soon as possible, right? So then we can combine these terms. We can move towards light, for example. So we can have something called positive phototaxis. We can move away from chemicals. So we can have negative chemotaxis. Okay, and then in combination, this ability is very powerful for microbes to be able to just kind of be in the area that they want to be in, right? Have some control. Okay, so let's move in now. Now we are in the cell wall. So how do I want to designate this? We are going to say yes. All bacteria, most bacteria, have a cell wall, right? So, right now we're just going to say all bacteria have a cell wall, right? So this is now a structure that's the same for all prokaryotic cells, right? And of course, for us, we mean bacteria, okay? We will learn a little bit later, again, exceptions to the rule, right? Cell walls are used to protect from the environment. It gives bacteria the shape, right? Maybe let's write that in there because I don't know if that's written anywhere, right? So it gives us the shape. So, hey, what shape is this cell? No, let's use the correct term, right? Yeah, bacillus, right? Good. So this is a bacillus cell, right? So the reason that this cell is bacillus shaped is because the cell wall has been built in this bacillus shape, right? And then as we learned from module 1, bacteria have cell walls made of this thing called peptidoglycan. Let's say that together real slowly, right? Break it down. Peptidoglycan, right? Peptidoglycan, right? Now, aka murine. But we're just going to call it peptidoglycan. Peptidoglycan is this, it's a polymer, right? Composed of these things called NAGs and NAMs. You can see the words right there: N-acetylglucosamine, N-acetylmuramic acid. If you want to memorize that, great, go for it, right? But NAG and NAM is fine, right? And, to be honest, if you just say that it's these carbohydrate backbone, that's okay too, right? So we have these carbohydrates then that run along this way, right? And then linking those carbohydrate backbones together are these short peptides. Remember what peptides are, right? They're proteins, right? These short chains of amino acids. Not quite long enough to be proteins, right? But they're short, usually 5, 6, 7 amino acid residues, linking them, right? So that provides this really nice, strong, sturdy structure that helps protect the cell from physical stresses, right? And oftentimes, if you can break these structures, if you can break the linkage there or break the linkage there, the cell starts to lose integrity. Now it's kind of only dependent on the membrane and often leading to cell death, right? So, the peptidoglycan layer is very important for bacteria and its cell integrity. The peptidoglycan is arranged around bacteria in 2 major ways, right? And we call these things gram-positive bacteria and gram-negative bacteria based on how they're colored using this staining procedure called the gram stain, right? And I think it's chapter 2 in your book, which we didn't cover. We'll go over the gram stain. If you're interested in it, it's a really good read. You go ahead and read it, right? But we're just going to look at how it's put together, right? So, and again, in the broad stroke, here we have the plasma membrane, which we'll define. This is the cytoplasm. So let's write that in there. This is the inside of cells, right? Inside of cells. And then this, of course, then would be the outside of cells, right? So gram-positive bacteria have many layers, and each of these would be a layer of peptidoglycan, have many layers of peptidoglycan smooshed together on top of the plasma membrane, right? Now, many layers are great. It sounds great, right? But the problem is that, all these layers then are able to go and move side to side, right? Kind of not making as stable as we want. So what they actually have are these things called teichoic acids, kind of linking all these things together. So we can have teichoic acids that link the layers of peptidoglycan together. And then the teichoic acid also anchors peptidoglycan to the plasma membrane, right? So now all of a sudden we have a really strong, rigid structure of peptidoglycan on top of our gram-positive cell wall, right? This gram-positive bacterium. Our gram-negative cell wall slightly different, right? So a gram-negative cell wall has once again, inside of the cell, plasma membrane has a very thin layer, 1, maybe 2 layers of peptidoglycan, right? And then it has a second membrane, right? So the space in between these two membranes is called a periplasm, right? Sometimes called the periplasmic space, right? But the periplasm is a space in between the two membranes, what we call the outer membrane and the inner membrane or cell membrane, right? With just a thin layer of peptidoglycan. And this provides vastly different function and characteristics to this type of cells, right? So gram-positive and gram-negative cells have vastly different characteristics. Embedded within the outer membrane is something called LPS, or lipopolysaccharide, right? It's a protein with a lipid moiety, right? We talked about lipoproteins. So we'll talk about that in a little bit and its function, right? But again, vastly different way. And so gram-positive and gram-negative cells have greatly different characteristics. So if we're going to go very quickly and say features of the gram-positive cell wall, we would say thick, many layers of peptidoglycan outside of the cell membrane, and then contains teichoic acids, which are unique to our gram-positive cells or our gram-positive cell wall. Whereas our gram-negative cell wall has a very thin layer of peptidoglycan in the periplasm, which is that space between the plasma membrane and the outer membrane, right? And that outer membrane contains something called LPS, which is unique to gram-negative bacteria. So let's talk about this outer membrane a little bit because it is important, right? The outer membrane helps protect the cell from the immune system, from antibiotics. We'll talk later about the flow of molecules through membranes. Membranes are really good at keeping things out, including things like antibiotics. And most antibiotics like to attack either inside the cell or the cell wall itself, right? So the cell wall, in this case, the peptidoglycan, is protected in some sort from the antibiotic attack because of this outer membrane. Furthermore, inside this outer membrane is this thing called LPS, or lipopolysaccharide, which is made up of lipid A and then this O antigen, right? And what the lipopolysaccharides can do is they function as this endotoxin during infection, right? So actually what happens is that when you're infected by a gram-negative bacterium, as the cells start to die, we're killing them off either with antibiotics that are effective against gram-negative bacteria or through our own mechanism of destroying bacteria, the cells start to release this LPS, which then causes this secondary issue, this inflammation response where it leads to things like fever, hemorrhaging, and then sometimes even shock, right? So sometimes when you have a gram-negative infection, you actually have to be careful how you treat that because you don't want to kill them too fast, right? Because of the fact that this LPS release is going to cause this secondary issue. Okay, so like we said, all bacteria have peptidoglycan, and then like we said that it's not always true, right? So there are other types of cell walls that exist amongst prokaryotic cells. Maybe the most significant of these would be the acid-fast cell wall, and it's called that because you can use this acid staining procedure, right? And these are ones that are able to still be stained using this acid staining procedure. And again, chapter 2 will tell you all about that, right? Really good stuff. Very significant because of the fact that acid-fast bacteria, the causative agents of things like tuberculosis are acid-fast bacteria, right? So this is often how we screen for tuberculosis. We take a little sputum sample from your lungs, do an acid-fast stain, and if you see these chains of bacteria like this, right, colored in red, right, those are our acid-fast bacteria. The acid-fast cell wall is this waxy lipid we call mycolic acid, right? So it's this really kind of waxy, almost looks like someone dripped candle wax when you grow these things, right? This waxy layer. And because it's so hydrophobic, it really helps protect cells during infection, right? And so our acid-fast bacteria then they look like gram-positive bacteria, right? Thick layer of peptidoglycan. And then this layer of mycolic acids is kind of sticking on top, right? This waxy lipid. Archaea, we said that they are prokaryotic. They don't have peptidoglycan, and that's true, right? They have other structural molecules. So for them, there's not really a uniform way of putting together cell walls. They'll have something called pseudomurane, right? Remember, murine is a word for peptidoglycan. They even have something called pseudopeptidoglycan. Some of them don't even have cell walls. They don't care. They don't want cell walls, right? So they have these different ways of kind of protecting themselves from the environment. And finally, mycoplasmas, which is a group of bacteria, of which the biggest genus is Mycoplasma, which then would be written capitalized and in italics. They are the smallest known bacteria. They don't even have cell walls, right? So these super tiny bacteria, they don't have cell walls. And in fact, they have things called sterols in membrane to provide strength, which is weird because sterols are a eukaryotic feature, right? Prokaryotic membranes don't have sterols, whereas eukaryotic membranes do. So again, there's always exceptions to the rule, okay? All right, so let's continue our journey. Let's move into the cell, right? And then the next layer, the plasma membrane. And this one here, what did I say last time? Yes, right? So we're going to say yes. All prokaryotic cells have a plasma membrane, right? Because remember, it's one of the features of cells. All cells have plasma membrane. And it's this thin layer that encloses the interior of the cell. In many ways, it's the border of the cell, right? And in fact, you can take a bacterium, you can strip away the peptidoglycan. If you do it really gently, right, the cell can still remain intact with just the plasma membrane itself. We already talked about this, made of phospholipids, arranged in this lipid bilayer, which we did not talk about, but we will right now. Right, so this is what a plasma membrane looks like, right? So we talked about this whole idea of this amphipathic phospholipids, right? Hydrophilic heads, hydrophobic tails, and then in this case, they're arranged in this lipid bilayer, right? So we have our hydrophilic heads facing the aqueous or water environment outside, hydrophilic heads facing the aqueous environment on the inside, and then our hydrophobic tails all kind of bunched together in the middle, kind of hanging out together, right? Away from the ugly water, right? Because it hates the water. So we have this mass layer of mostly phospholipids, right? Embedded within the membrane are proteins, right? We call them integral proteins because they are kind of separate or connect the outside to the inside, right? And they perform various functions. They can have channels that allow things to move. They can have transport proteins, which will move things in directions that molecules don't want to move. We can have structure, right? Things to provide strength. We can have transport and all this kind of stuff. You remember this whole glycolipid stuff, right? We can have glycolipids or these carbohydrate moieties stuck to outside, and the cells will use these things to talk to each other, all this kind of stuff. And the important thing here is that the phospholipids, they're not static. They're constantly in motion, right? So it's referred to as the fluid mosaic model. These things are always kind of swimming around. And in many ways, I like to think of this as like an ocean, right? So here you are looking out in the ocean, and these little things are just kind of bobbing and floating around on the surface of the ocean, right? With a whole bunch of good stuff going on underneath. All right. So I kind of alluded to the fact that the membranes are very good at keeping things in and out, right? And so we actually say that the membrane is a selective barrier or has this selective permeability, right? So permeability means whether things can pass through or not, right? So to be selectively permeable means that only some molecules can pass this barrier, right? So here is what cannot. Large molecules cannot, right? They're just too big, right? They just can't squeeze through the membrane. Ions cannot, right? Remember, phospholipids carry charges, right? Phosphates are negative charge. So ions are repelled by the charges on the phospholipids. And actually, they're more than just negative charges. There are also some positive charges, right? So in just general, ions cannot. However, what can pass are small hydrophobic molecules, right? So things like oxygen, carbon dioxide, they can pass through because the core of the membrane is hydrophobic, right? So all these things need to do is kind of slip past the hydrophilic heads, and then it just moves around across to the other side, right? So oxygen, carbon dioxide, they pass quite freely through membranes without too much of a bother, right? Oh yeah, let's define what a large molecule is, right? So something like glucose, 6 carbons, Glucose cannot pass, right? So glucose and bigger cannot pass, okay? Of course, something like glucose needs to come into the cell, right? So for this case, we have transport proteins that allow molecules that normally cannot pass unaided, right? So things like ions, things like glucose, we do need them to cross, right? So we can then regulate this using transport proteins, okay? So how do things cross membranes, right? Transport across membranes is extremely important. And we have two general processes, two general ways that these things move, right? We have what's called passive processes and active processes. So let's define a passive process, right? Let's use the long term first. Molecules move from an area of higher concentration to an area of lower concentration, or something we call down the concentration gradient. So, I always like to draw a graph, right? And we have concentration, high concentration to low concentration, right? Over time. So we're moving things down the gradient, right? So now we're going to take this graph and imagine this is actually a hill, and we're going to place a rock just like that on top of there, right? What is going to happen to this rock, right? Well, we are all reasonably intelligent people, right? We know that this rock will roll down the hill, right? And how much energy does it take for this thing to roll? The answer is none, right? So passive processes require no energy, right? It just happens, right? So we're just going to let nature do what it do. And this is going to keep going until the rock gets to the bottom, right? So once it's at the bottom, now the molecules are going to stop, okay? Basically, there's no more gradient. And we'll talk about that in a little bit. An active process is the opposite, right? So now we have, again, concentration gradient moving from an area of low concentration to an area of high concentration, right? So, or what we say up the concentration gradient. Well, once again, we're going to stick a rock up there. And if we want to move this thing against the concentration gradient or up the concentration gradient, what do we require? Well, now we know it's going to require some energy. You're going to have to push this thing a certain way, right? And the bigger the gradient, right, the steeper the slope, the more energy is going to be required, okay? So let's keep that in mind as we talk about the specific mechanisms, right? And the simplest way for molecules to move is this thing called simple diffusion, right? So diffusion is simply the movement of molecules from an area of high concentration to an area of low concentration, right? Down the gradient, right? So we can then have a system where, or situation where we have our membrane, and we're always going to talk about moving across membranes. So we have a membrane here. In this case, this will be outside, this will be inside, right? Where's our concentration gradient? High concentration gradient outside, low concentration gradient inside, right? It wants to simply move down the gradient. So in this case, we're going to move from outside to inside. So things are going to start flowing through until we get to this point where we're balanced, right? Or it's equal, right? And that's what we call equilibrium. So at the point where we're equal, we have no longer a concentration gradient. Now our net movement is going to be 0, right? The movement, the molecules, they'll still move in, they'll still move out, right? But the net movement is going to be the same. So we're going to maintain this concentration gradient. And this is really important for small hydrophobic molecules that can move across membranes freely, right? Remember, they can move across membranes freely. So things like oxygen and carbon dioxide, right? So imagine then this is oxygen. This is kind of how it would work. We breathe in oxygen, right? So we have a bunch of oxygen out here. And then oxygen is going to move into the cells down the gradient. And then what we're going to start doing is once it's in the cell, we're going to start using the oxygen. So the oxygen starts to disappear, right? And so what ends up happening is that we maintain a continual gradient of high oxygen outside, low oxygen inside, right? So this oxygen is going to flow into the cell. And the opposite is true for carbon dioxide. We're going to produce carbon dioxide in the cell, and we want to get rid of it. So the carbon dioxide is going to flow out, right? And then the blood's going to take all that carbon dioxide away. So we're going to maintain this continual gradient of lots of carbon dioxide on the inside, very few on the outside. So then the carbon dioxide will just leave the cell, allowing us to continue our respiration. Sometimes we want to move things like the food that we eat into cells and out of cells, but we can't, right? Because remember, large molecules don't cross easily, right? But at the same time, we don't want to use energy. Using energy to move things like food to make energy is silly and wasteful, right? So what we have in this case is something called facilitated diffusion. This is, again, movement down the concentration gradient, takes no energy, right? But it's for molecules that can't cross membranes, things that are large like glucose, and things like ions, right, that cannot normally cross the membrane by themselves. So in order for this to happen, we need help, or we need to facilitate diffusion to happen, right? So this requires transporter proteins, integral membrane proteins that are specific for certain molecules. So in this case, let's imagine that this is glucose, right? This is actually the shape of glucose. Here we have a concentration gradient of lots of glucose on the outside and then very few on the inside. We have this transporter protein, which is basically a hole that fits glucose. And then this allows glucose now to flow into the cell and make its way in. And because it's moving down the gradient, this requires no energy, right? And again, just like oxygen, once it's in the cell, we are going to start eating these things. So we'll maintain this perpetual gradient of glucose always wanting to flow down. Okay, and this is actually a really neat system, because if the cell is full and we say, you know what, we don't want any more glucose to come in, all we need to do is close off the hole. Now glucose won't come in, right? Even though it wants to, but it won't, right? Because it can't cross the membrane. And then we can then say, okay, cell, you're done eating for a while. Let's use up this energy. And then we can go and open it up again when we're ready, right? Again, requires no energy, just like simple diffusion, right? But it requires a transporter protein because these molecules can't flow naturally. And again, we can have the opposite situation where we could have things like ions and stuff that want to flow out, right? Down the concentration gradient because there's too much on the inside, not so much on the outside. And then we can open these channels and just let them go. All right. Sometimes we need to move things against the gradient. And we'll talk about one of these very important things. One of the ways we make energy is to move things against the gradient, right? But this is called active transport, right? So active transport requires energy, right? And you guys remember what this currency of cell energy is? That's right, it's ATP. So most active transport requires ATP for it to work, right? So here we have this sodium channel, right? We have a bunch of sodium on the outside, right? Very few on the inside, right? But this cell, it wants the sodiums out there. It needs to get them out, right? And for those of you who have studied things like muscle contractions, it needs those sodiums out, right? But it's going against the gradient. So in order for that to happen, we need to use up some ATP. So every time ATP comes in there, a bunch of sodiums get spit out, right? And it requires this constant supply of energy. It requires this constant flow of energy. Same with potassiums. We got a bunch of potassiums on the inside, right? Some on the outside. We need to pump it against the gradient. We're going to require some ATP to power this process, right? So it requires transporter proteins, requires ATP. Doesn't always require ATP. Sometimes we can use the energy in food and light, and we'll learn all about that in module 4, I believe, where we can use the energy in food and light to power active transport, right? But this is movement against or up the concentration gradient. Requires energy, requires a transport protein. Okay, we have a special situation where we're talking about the movement of water, right? And the movement of water is called osmosis. Sometimes people say osmosis is like diffusion. Sort of, I guess, right? But they're actually very different processes, right? We're talking about movement of water down a concentration gradient, right? So it is like diffusion in that, right? And we already know then this requires no energy, right? Because it's moving down a concentration gradient. But we're also talking about movement across membranes, right? So osmosis is the special case. We're not talking about just movement of water. We're talking about movement of water across a membrane. Now remember, water can cross membranes, no problem, right? Solutes cannot, right? So really what ends up happening is we have this situation where we have unequal solute concentrations, which then would mean we have unequal water concentrations, which then causes the water to move. Because the water can move, solutes cannot. All right. And so then this brings us to this term called osmotic pressure. This osmotic pressure is a pressure that's caused by changes in solute concentrations on each side of the membrane. Okay. So if the pressure or the differences in concentrations are too great, we're going to have great osmotic pressure. All right. And this has an effect on the cell. All right. And so this measure is something we call tonicity. Right. It's a measure of solute concentrations relative to a cell. So let's walk our way through the three scenarios, right? So we have something here called an isotonic solution, right? And iso means equal. So here we have a solute concentration outside that's equal to the solute concentration in a cell. Remember, solutes don't cross, right? So if the solutes outside are equal to the solutes inside, right? Let's draw this, right? So let's have something where we have 10 parts. Okay, so we can have, let's say, 7 solutes. Oops, I'm gonna cut that backwards. That goes 7 water molecules for every 3 solute molecules. Okay, and inside the cell we have 7 water molecules for every 3 solute molecules. Okay, so if the solutes on the outside are equal to the solutes on the inside, that means the water on the outside is equal to the water on the inside. All right, so that means we are at equilibrium. Water will flow in the same rate that it flows out. So there's no net movement of water across membranes. And isotonic situations are what cells want to be in. They're happy in isotonic situations, right? Not always the case. Sometimes we have something called a hypertonic, right? Hyper means great, right? So now we have great solute concentration. Solute concentration is higher outside, right? So let's draw this now. Let's go same cell, right? But now we have 5 solute, right? And remember 10 parts, that means we'll have 5 water, right? So then inside the cell, we're gonna, what if we say we have the same cell? 3 solute, 7 water, right? So now we have our solute concentration is greater outside than it is on the inside, which then means that our water concentration outside is less than our water concentration inside. So now we have this concentration gradient. Water wants to move from high to low. So the movement of water is going to be outside the cell, right? It's going to go from inside to outside. And it leads to something called crenation, which is this shriveling of the cell, or when it gets real bad, something called plasmolysis, where the water loss is so bad that our cytoplasm has essentially shrunk to nothing. And this is bad for the cell. In fact, this is one way that we can preserve our foods. Hypertonic environments, right? High salt environments. We preserve our foods using high salts because of this effect, this plasmolysis effect on bacteria. And we'll talk more about that in a few chapters from now. A few modules from now. Yeah. Okay, so hypertonic solutions, cells hate it. Sucks, right? Because of the fact that it starts to shrivel. In fact, it's the reason we can't drink salt water, right? Salt water would be hypertonic to our cells, right? If water is too salty, our cells are going to start losing its moisture, right? Losing the water inside, and it's going to start causing our cells to shrivel or plasmolyze, right? It's the reason that you shouldn't be eating too much salty food, because of the fact that our cells will start to get dehydrated through to the loss of water, right? Overall causing this imbalance and then poor metabolism. The opposite of this hypotonic. Hypo means under or lower, right? So let's say in a hypotonic situation, right, we have, let's say, 1 solute outside, which then would mean we have 9 water outside, right? And then inside the cell, again, same cell, we have 3 solute inside, which means we have 7 water, right? We have less solute outside, which means then we have a higher concentration of water outside than inside, right? So now we have our concentration gradient will be from high outside to low inside, and then water will start to flow in, right? And if this osmotic pressure is great enough, right, there's so much difference that the water just rush it in, the cell can burst, right? Something called lysis, right? Osmotic lysis, right? It's this bursting of the cell due to the fact that there's tons of water rushing in. Bacteria in general don't mind hypotonic solutions. They don't like it, right? It still affects their metabolism, right? But they survive, right? They have this cell wall that counteracts this osmotic pressure. So for the most part, they don't burst, right? And gram-positive bacteria, they don't really care about this too much, right? Because the cell wall protects them. But our cells are not so good, right? So we don't like that, right? So there's something that can happen when you drink too much water, right? In fact, I had a colleague who drank so much water that she ended up going to the hospital, right? Because you're creating this hypotonic situation inside you, right? Now we're going to create this imbalance. Water's going to move into the cells. Cells don't like it, right? And then so causes a lot of problems, right? So hypotonic solutions are also bad. Isotonic is what we want. Okay, let's move into the cell now, right? And here, cytoplasm, yes. All cells have a cytoplasm. Basically, it's just a fluid inside the plasma membrane. It's mostly water, about 80%. And then the good stuff, proteins, carbohydrates, lipids, even DNA, nucleic acids, ions, all these things kind of floating around in solution, right, as they're moving and whipping and interacting with each other. And then some specialized structures, right? But all cells have a cytoplasm, as we discussed, right? So let's move into the cell now. And then notice there's not a lot of stuff going in there, just some things. So we'll talk about some of these things. The nucleoid, answer is yes. All bacteria have a nucleoid. Basically, it's the region that the chromosome hangs out, right? The chromosome is large. It's very big for a cell, right? It's got to be packed. So they have these things called nucleoid-associated proteins, or NAPs. We all have our NAPs that help organize and pack this chromosome, right? Stuff it into this nucleoid. Oh my goodness, I can't believe I said nucleus. Stuffs it into this nucleoid, right? But basically it's this general area where the chromosome is hanging out. And it's not really anchored anywhere. It can sort of move around, but it's sort of together, right? Some bacteria have these things called plasmids. They're not part of the nucleoid. We will talk more about plasmids a little bit later in module 7, right? They're not part of the chromosome. They carry vital information. They just float around in the cytoplasm. Okay, but the nucleoid is a region where the DNA molecule hangs out, right? And it's organized by NAPs. Moving in, ribosomes. The answer here is yes, because remember, one of our criteria, all cells have ribosomes, right? So the ribosomes are responsible for making proteins. And remember how important proteins were, right? Proteins are cell tools. So much of our cell is made up of protein, right? So ribosomes are vitally important, right? It is one of the defining things for a cell. The ribosome has many subunits, right? It consists of proteins and ribosomal RNA, or rRNA. Remember we talked about RNA last module, right? And then we said there are 3 types of RNA. One of them was rRNA, ribosomal RNA. Well, this is where you find them, right? So these ribosomes are this mixture of proteins and rRNA. There are many subunits to this thing, a lot of pieces, right? But the 2 major ones are what we call the large subunit and a small subunit. And they're called that because one is big, one is small, right? The bacterial ribosome is what we call a 70S, because those of you who are super clever will see that 50 plus 30 is 70, which of course is not true, right? But that's basically a rounding error, right? Large subunits are a little bit less than 50, so they call it 50s. Small subunits are a little bit less than 30, so they'll call it 30. But when you put the two together, it's a little bit less than 70, so we just call it 70. And the S simply refers to this unit of size. We don't want to get into that, right? But the bacterial ribosome is often referred to as the 70S ribosome because of its size. And that's important because eukaryotic ribosomes, as we'll learn a little bit later, are a little bit larger. They're 80S ribosomes, right? And we can actually use this difference, the difference between the 70S and this 80S ribosome, to target our antibiotics, right? So we can find, and there are many antibiotics that attack specifically the 70S size, not the 80S size. We can then inhibit bacteria, but we will be okay, right? So that's often many ways that we can then have therapeutics. But in general, ribosomes make proteins, right? 70S. Good. Inclusions. You guys want to guess? Yes or no? Not, right? So inclusions are not found in all microbes, right? So inclusions are these structures that are inside cells that can be used for, let's just call it various functions. And really it depends on the type of inclusions, right? So when you look at these things, we have deposits, right? Things like this. So some of these can be sulfur granules where they require and they eat sulfur. So they need a little bit of extra. In times of plenty, they'll just store it. It's many ways like they're fat deposits, right? Same goes with phosphate, same goes with carbon, right? So something called PHB is a carbohydrate, right? That they can then store inside. And PHP is interesting because it's wrapped in a phospholipid monolayer. And so in many ways, this is kind of like an organelle. And later we'll learn what organelles are, right? So PHP are like a pseudo-organelle, right? But not a true organelle. But basically, these inclusions can be used to often store nutrients, but sometimes can do things like detect magnetic fields, detect light energy and stuff like that. So it allows it to react and move to things, right? So inclusions are sort of like organelles, not true organelles, right? But again, not all microbes have them. And then endospores, guess, yes or no? Not, right? So again, only some microbes have them, right? The definition of an endospore always is specialized resting structures, whatever that means, right? Formed by some bacteria. They're typically gram-positive bacteria. Let's write that in there just for fun, right? So gram-positive bacteria. And the two most significant genera are Bacillus and Clostridium because they contain some hardcore pathogens. Things like Clostridium tetani, guess what disease that causes? Tetanus, right? Bacillus anthracis, guess what disease that guy causes? Anthrax, right? So some really fun diseases that these things cause. Gangrene, lots of good stuff, right? And it's this endospore that allows it to survive these various environments, right? So by a resting structure, really what we mean is, I like to call it like a cell inside a cell. So here we have this this cell, right? This would be our bacillus cell. And then inside we got this large resting structure, right? The structure that's inside, that's essentially a copy of itself within, okay? This thick protein layer is resistant to things like drying out, heat, chemicals, UV radiation, right? So these really extreme environments. So really then what happens is that when the cell dies because things get too hot or too much UV light is shining on it, all these cells, oh no, they're dying. Right, but the endospore will be able to survive, right? So it'll be able to live. It's metabolically inert, right? They can survive for literally millions of years. Scientists have recovered endospores that are millions of years old, right? So when times return to normal, they germinate, which is return to this vegetative or actively growing form, right? So it's very important in the food industry, right? Many of the things that cause issues in the food industry, mostly botulism, right, which is a food industry disease, is caused by Clostridium botulinum, right, which is an endospore former, right? It's and then this endospore, if you don't properly treat your food, right, the bacteria might die, but the endospore will kick around, right? And when that thing germinates after you close it off in the can, well, then it's going to go and come back to life and then give you some serious disease. Okay, if you want to take a little break, now will be a good time because we are going to move on to our eukaryotic cell. In fact, I'm going to take a little break to deal with this text message that I'm getting, right? So let's take a little break and then we'll come back. If you want to keep going, you can keep going because for you, it'll feel like I did not take a break. So let's move on to our typical eukaryotic cell, right? And the key word here I want you guys to focus on once again is the word typical, right? Just like with our prokaryotic cell here, they got a really lovely picture of a cell, right? But you're not going to see cells that look like this, right? But they're trying to show you what most cells kind of look like, right? Typically, our eukaryotic cells are larger, more complex than prokaryotic cells. And in general, the complexity will be outside the cell. So the outside of the cell will be much more, inside the cell. The inside of the cell will be much more complex than the outside of the cell, which tends to be a lot more simpler for eukaryotic cells, right? Because the good stuff is happening on the inside. Particularly these specialized compartments that we call organelles, right? And the word organelle simply means mini organs, right? Teeny tiny little organs that have specific functions for eukaryotic cells, okay? Like with bacteria, right? A lot of different morphologies or shapes, right? Eukaryotic cells have a lot of, just like bacteria, no, probably more than bacteria, a lot of interesting shapes. Many of them are irregular. So we don't often classify them as things like coccus and bacillus, right? Just because the shapes are really specific to function. Here's a skin cell with a really weird shape, right? Sometimes they'll be round, sometimes they'll be kind of this oval or what we call amoeboid, right? But it's often the shape is often dictated by the environment that they're in or the functions that they need to do. Okay. So just like with our last section, we are going to move from outside to inside. We will indicate which structures are the same, which ones are not. I don't know what I was going to write there, right? So not, right? So flagella, cilia, and we actually saw flagella before, right? So do you guys remember what flagella do? Yeah, used for motility, right? So flagella are used for motility. It's different type of motility. They work differently than they do in bacteria, right? So in bacteria, remember they do that whole rotating type of thing, right? Here, this is more like a whip, right? So in many ways, like when you are doing like a front crawl, right? And then you're kicking your legs. In many ways, that's kind of how flagella work. They just kind of whip around and the thing swims through, right? They can be arranged in many, many different ways. Eukaryotic cells can have one single flagellum. They can have multiple flagella, but it's this whipping motion that kind of drives them through. And in this case, motility is a lot more directed, right? So they kind of move to where they want to move based on the environments that they're sensing. Cilia, on the other hand, cilium for singular, cilia for plural. They're these short little hairs that cover the surface of the cells, right? And they are used for motility. They can be used to move. So they do like this weird wave-like motion where they're all kind of waving in the same. So like if you were to take your hands by your side, you just kind of flap them up and down, right? So they can move using cilia, right? But they can also use to move things across cells, right? So just kind of sweep materials across cells, right? So our respiratory tract cells have cilia, right? So when things get stuck down deep in our respiratory tract, we can use these cilia to move them up and out to make sure they don't get down into the sensitive areas of our lungs. Cell walls, yes or no? Answer is not, because remember, not all eukaryotic cells have cell walls, right? So they're specific for the type of cells that we have, right? So cell walls generally much more simple than they are in prokaryotic cells, just like basic polymers of a polysaccharide, right? So it's not this complex linked polypeptidoglycan-like pattern, right? We've already learned that plants and algae have cellulose cell walls. Fungi have chitin cell walls. Both of these are actually just polymers of glucose. But like prokaryotic cells, cell walls provide structure, they provide protection, right? But many eukaryotic cells don't have cell walls. They do just fine without it, but then they're kind of subject to some of the stresses that life without a cell wall has. Extracellular matrix, it's not really a structure, but it is kind of an important thing, right? So we'll talk about it. Basically, it's the stuff outside of the cell, right? And so eukaryotic cells, right, they are much more social than bacteria, right? So they need to respond with their environment. So what they do is they spit out this sticky mass of carbohydrates, of proteins, and that just kind of floats in the space outside of the cell. So if it's by itself, it'll just be kind of surrounded with this extracellular matrix that it hopes to like grab things with and use to like poke and sense the environment. When we are in this multicellular organism, this stuff connects between cells, right? And actually one cell can tickle one end of its extracellular matrix protein, and then that signal can make its way through to another cell, and then this first cell will then tell the second cell what's going on. It can help a little bit with external stresses, right? Because of this layer that's around it, right? And then basically it's very important for cells, right? But not a true cell structure. Some pathogens, they're clever. They say, hey, look at all these glycoproteins and lipoproteins floating around, right? And they actually use this matrix to kind of stick around, right? So if you can get this bacterium that just sits right here, stuck onto this thing, what it's going to do is like whenever this thing sends out some sort of sugar or carbohydrate to go and send over to his buddy Steve over there, this bacterium will say, yoink, and take it and eat it, right? And so it's this really cool place to hang out and do some stuff and then steal some of the nutrients that are secreted by the cell. All right, moving in plasma membrane, we should know the answer to this, right? The answer is, of course, yes. All cells have plasma membranes, and the eukaryotic plasma membrane is incredibly the same. Okay, so they're pretty much the same as prokaryotic membranes. They have these phospholipid bilayer, like we talked about, fluid mosaic model, like we talked about, proteins, like we talked about, semi-permeable layer, right? Big stuff, no. Ions, no. Small hydrophobics, yes, right? One major difference is that the eukaryotic membrane has sterols, right? So remember one of our lipids, the sterols, stuck within the membrane, and that helps provide a little bit of extra strength. Because remember, a lot of eukaryotic cells, they don't have cell walls, right? So the sterols kind of prevent things from moving too much, help give it some strength and rigidity in times of stress, right? And then it can also be used to help communicate between. And in this case, eukaryotic cells are very much dependent on things like glycoproteins, glycolipids, right? These carbohydrates that they kind of have stuck on the outside that they use to talk to each other, right? And in fact, I think did I mention this already? Often things like viruses say, hey, this is a perfect place to stick. And so many viruses and even some bacteria will use these glycoproteins to go and stick and then enter cells. Speaking of entering cells, right? Stuff on the outside needs to come in, stuff on the inside needs to come out, right? We have this thing called endocytosis. Right, and what endocytosis is this uptake of materials through membrane invagination, right? In many ways, if you want to think about this, it's sort of like creating this temporary mouth that swallows things, right? Two major types. Phagocytosis is this ingestion of large particles, right? What we call cell eating. So this is what immune system cells do to get rid of bacteria. Here we have a bacterium. And then the immune system cell will stick to it, invaginate it, and pop it into this thing we call a vacuole, right? And then now it's kind of like stomach, where then it can then digest it. And we'll talk about that later. Pinocytosis are small particles, small little things then that once again stick, we invaginate it, and then we pop it into, in this case it's called a vesicle. Small things, small molecules, right? Viruses have hijacked this mechanism, right? So viruses say, hey, we're going to stick to the outside, and they can get into the cell through pinocytosis or overall endocytosis, often through this thing called receptor-mediated endocytosis, where rather than just kind of drinking it or eating it, our molecule first sticks to the membrane via this receptor, and then that thing gets popped in or invaginated just like in a vesicle. And of course, things that come in at some point needs to go out, right? So there's something called exocytosis. What's the opposite, right? So in this case, we'll have our vesicle or vacuole. It'll come out this way, and then we can excrete materials to the outside. Okay, so that's the general flow for things going in and out for some types of molecules. Ribosomes. We heard of these before, right? Yes, all cells have ribosomes, just like with prokaryotic cells, right? Ribosomes. Hey, look, I didn't even write it on here, right? So what do ribosomes do? Yeah, ribosomes make proteins, right? So vital to life, right? All cells have them. In the eukaryotic cell, we have two types of ribosomes. Ones that are free floating in the cytoplasm, so they make proteins that are intended for being in the cytoplasm. And then we have membrane-bound ribosomes, typically attached to the ER, or the endoplasmic reticulum, which we'll learn about in a few slides. And these make proteins that are going to be attached to the cell membranes, or sometimes for export out of the cell. And remember, ribosomes, they have their own DNA. That's not what I wanted to say. Ribosomes are, eukaryotic ribosomes are larger. They're 80S as opposed to 70S, right? Which are, again, this difference between the eukaryotic and eukaryotic and prokaryotic ribosomes, right? Whereas a mitochondria and chloroplast, they also have ribosomes. They also used to make proteins. They are 70S ribosomes, just like bacteria, right? But they really only make proteins that are specific for the mitochondria and the chloroplasts. All right, nucleus. I think I don't know if I need to stop this, right? We're pretty much going to say everything's going to be S, right? Because there's stuff on the inside. So the nucleus is where the DNA is stored, right? It's where our genetic information is stored. Not a single lipid bilayer, 2 lipid bilayers that surround the DNA that's on the inside. So multiple chromosomes. Remember, eukaryotic cells have linear chromosomes, right? So we have this double lipid bilayer called the nuclear membrane that surrounds the chromosomes on the inside, right? Various filaments called the nuclear lamina that maintains the shape of the nucleus, right? Kind of provides strength for the nucleus. And then there's this region within the nucleus called nucleolus, right? That's right here. It's this kind of a darker, dense region, right? This is where the rRNA comes from, right? So here are the genes that encode the rRNA, right? But in general, if we're just going to say broad stroke, the nucleus, double membrane, right, stores the cell's DNA. So this is where the DNA is found. The endomembrane system isn't an organelle. It's kind of a collection of organelles. So we're going to talk about each of these organelles separately, right? But there are a series of membrane compartments that are used to move things in and around the cell and sometimes out of the cell. They consist of the endoplasmic reticulum, the Golgi apparatus, lysosomes, and then the vesicles that kind of float in between. So let's talk about the endoplasmic reticulum, right? The endoplasmic reticulum are these series of flattened network of membranes that extend from the nucleus, right? And so actually what happens here is that the information goes from the nucleus, right? This is where our RNA is made, then moves through the endoplasmic reticulum, right? And then here we have that's called the rough ER. They're covered in ribosomes, right? Remember what do ribosomes do? They make proteins. So they make proteins in here that are going to be destined for either the plasma membrane, where they can hang out in the membrane, or we can ship them out and move them from one cell to another cell, right? So, and they're shipped in our transport vesicles, right? So it's these transport vesicles then that take the proteins that are made in the rough ER and then move it out to the membrane. And then we have the smooth ER that also extends from the nucleus. They're involved in doing things like making lipids. They're involved in some carbohydrate metabolism, as well as the detoxification of some toxins, right? So collectively, the endoplasmic reticulum is very important for making things, making molecules, right? Primarily lipids and proteins, right? But then it can also have some other functions as well. And it's this flattened network of membranes that extend from the nucleus. Just outside of that, is something called a Golgi apparatus, named after Dr. Golgi. And what it is it's also like this stack of membranes. Right? These are these stack of membrane discs called the cisternae. And the Golgi apparatus then takes proteins that it receives from the endoplasmic reticulum. It takes lipids that it receives from the endoplasmic reticulum. It takes carbohydrates that it receives from the endoplasmic reticulum, and then it adds things to them, right? Typically, it's glycosylated, which means it adds proteins, right? So, carbohydrates. So we'll add carbohydrates to these various things, and so this is where our glycolipids come from, our glycoproteins. Sometimes it'll add other types of tags to them, right? And so these are signaling molecules, and most of the things that are going to go through the Golgi apparatus are going to be exported out of the cell. Some of them are going to be inserted into the membrane, right? So this is where protein modification happens. So it's all about modifying and then transport of proteins. And then finally, the last thing in the endomembrane system are lysosomes, right? So lysosomes are these membrane compartments that contain many digestive enzymes. And so they're used to break down things like food, damaged organelles, cell debris, microorganisms, whole bunch of different things, right? So when an immune system cell, like we talked about phagocytosis and pathogen, a bacterial pathogen, it then uses the lysosome to break it down. If we have a cell that's dying and we need to get rid of it, we're going to ingest that, use lysosomes to break it down. If we have organelles that are now lost, it's well past its function, right? It's going to use lysosomes to break those things down, so then we can recycle those elements. Okay, so lysosomes, Golgi apparatus. I guess I kind of forgot to continue my pattern, right? But yes, they are contained in all cells. Peroxisomes, yes, right? Peroxisomes they're not part of the endomembrane network, but they're very similar, right? It's just this membrane compartment that helps break down toxins, helps break down amino acids, fatty acids, right? So it aids in the digestion of things and the recycling of elements, right? And then it also helps in the lipid synthesis, right? So actually, if you look at the functions, very similar to a lysosome, similar to the ER, right? But for some reason, we don't consider it part of the endomembrane network, right? But peroxisomes, we can say then, breaks down toxins, helps in the synthesis of some molecules. All right, cytoskeleton. Yes, right? The term cyto, or the prefix cyto, by the way, refers to cell, all right? So this is the cell skeleton, right? Made up of 3 major types of filaments called microfilaments, intermediate filaments, microtubules. You know, your book goes a lot more detail into this. Super important stuff. We're not going to talk too much about it, right? Because we are going to mostly focus on bacteria which don't have cytoskeleton. But the cytoskeleton provides support for cell functions, provides strength, transport, movement. In fact, when the cell moves, it's using the cytoskeleton to kind of move the cell body forward, right? When it does this pseudopod or this kind of walking, right? When it uses phagocytosis or endocytosis, it uses these cytoskeleton elements to just kind of wrap the membrane around things, right? So it's very important for many of the functions that happens, right? But I think this is about as far as we want to take it. Used for strength, transport, movement, made up of microfilaments, intermediate filaments, microtubules. Okay. Mitochondria. Yes, all cells have mitochondria, right? Because they're a very important place for carbohydrate metabolism, right? So this is where we're going to make ATP. Okay. Just like the nucleus, it's double membraned, right? In fact, it looks very much like a bacterium, has its own DNA, has its own ribosomes, right? But this is the site of ATP production, right? So all our ATP comes from the mitochondria, right? Mitochondria on singular. So we're going to get tons more detail into the function of carbohydrate metabolism, right? But for now, sufficient. Mitochondria is where we metabolize our carbohydrates and we make our ATP. Chloroplasts. Guess. Not. All right. So these are found only in photosynthetic organisms because this is where photosynthesis happens, right? Just to be better than the nucleus and the mitochondrion, 3 membrane system, right? So we don't need that 2 membrane stuff. If 2 is good, 3 is even better, right? So we have this outer membrane, this inner membrane, and a thylakoid membrane. And it's the thylakoids where our photosynthetic pigments are. It's the thylakoids where the energy is absorbed, right? Again, contains its own DNA, contains its 70 S ribosomes, right? So that whole endosymbiotic theory, right? You can read all about that again if you want to, right? But in general, chloroplasts, site of photosynthesis. And we'll learn more about photosynthesis in module 4. If you want a super short list of the organelles and their main functions, well, here it is, right? So I'm just going to leave it on here. You can pause it if you want. And, you know, this is probably get you through most of the quizzes that we do, right? You know, slight application of this, right? But here's a very quick laundry list. And this concludes the cell for us, right? So we are done with the cell, right? So now that we've gone through our biochemistry, now that we've gone through the cell, we are now ready to talk about some of the microbes in the microbial world, right? So we're going to do this general survey of some of the microbes, talk a little bit about some of the interesting differences between microbes, all in module 3. So we'll see you soon.